Good afternoon or evening, and welcome. Kensington Senior Living has 7 unique communities, 4 located in Virginia, Maryland, and New York, and 3 sister communities located in California. We are truly privileged to support care partners and professionals coast to coast. We focus on relationship-based care built on the foundation of our promise to love and care for your family as we do our own. That promise is what initially drew me to become a member of the Kensington family, as my father lived there for almost 7 years. All registrants today will receive a copy of the webinar to share with friends and family. Please also use the Q&A function to submit questions during our live Q&A. And today joining me is Dr. Omar Abdul-Rahman, and you're in for a treat as our event is titled, Know Your Genes, Empowering Families to Understand Risk and Treatment of Genetic Conditions. Omar believes, as a clinical geneticist, and I probably just botched that, you're gonna have to help me, Omar. My role is to help families affected by a suspected genetic condition to get answers regarding what the condition is, how it occurred, and what we can do to best support the affected individual. Knowledge is empowering. I'm so glad you could join us today, Omar, and thank you so much for your time. And we're super excited to hear about genes. Everybody has them. This kind of affects everybody. So I'll let you take it from here. And then I'll be joining you when finished for the Q&A. And again, please use the Q&A function to our audience. Thank you so much.
Thank you, Susan, for that kind introduction. As Susan mentioned, I am a clinical geneticist, and I got to that route by initially going to medical school and completing a pediatric residency at the University of Mississippi. I then went to California, where I did my genetics fellowship at Stanford University. And in genetics, because it affects all ages, we learn lifespan. So we learn how to take care of pregnant women who might be experiencing a genetic condition identified in their baby. We deal with children who have various genetic disorders, but we also help and diagnose adults with various genetic conditions. Some of those might be cancer related, some of them might be late onset types of conditions. So we really do deal in lifespan. And this is a very exciting time in genetics because there's become much more available genetic testing, both to support physicians, but also towards the general public. So I wanted to kind of give you a little bit of context of what's happening in the genetics world. So to start, let me first get my slides up. So you should be seeing this mouse moving over here. So I'm just going to start off with this picture because, as Susan says, we all have genes. We all know a little bit about genes. But it's really remarkable that it took so long to figure out what was really driving heredity. So this is a picture of Reese Witherspoon and her daughter. And I think almost anyone would say, gosh, they look a lot alike, which makes sense. But as a clinical geneticist, we are also trained to look for very subtle variations in how people look, because there are a lot of genetic syndromes that are diagnosed based on that. I think most people would be familiar with something like Down syndrome, but there are over 5,000 different genetic syndromes. So when I look at this picture, for example, I see the similarities, but I also see some subtle differences. So for example, you might look at the corner of Reese's eye and notice that it comes together almost like the top of a teardrop, whereas her daughter's doesn't. It kind of looks more like the end of an almond, if you will. And if you look at her father, Ryan Philippe, you'll notice that he too has a more similar almond shape to his eye. The other thing I notice, for example, is the nose. While they have a very similar nose, actually kind of hangs over a little bit, has this little extra bit that kind of hangs down compared to out here. Whereas her daughter does not seem to have that. Hers seems to be a little bit flatter. And again, if you look at her father, you'll notice that that's where that came from. And the third thing that stands out to me are the lips. if you look at the lips and you look at the volume of the upper lip to the lower lip, you'll notice that Reese has approximately an even distribution of volume to her upper lip and lower lip. But if you look at her daughter, you'll notice that there's a bit more volume here in the lower lip. And once again, that's something that's really mirrored from her father. And so you can see in this particular example that people are really a combination of their parents' traits. And those traits, we finally learned in the early 50s, are transmitted through this molecule called DNA. And DNA is present in every cell of your body. And it is essentially the code for how information is transmitted from one generation to the next. And that code is written in a very simple alphabet of 4 letters. Those 4 letters are A, T, C, and G. And so it really is a little bit mind-blowing when you think that you can write out code to really produce a human being with just those simple 4 letters. It's really quite remarkable. Now, in order to contain all of that DNA, we know that that DNA is quite long and it has to be packaged into each cell. Otherwise, it would just be all over the place. And so the DNA molecule is actually tightly wound into structures known as chromosomes. So chromosomes are just packages of DNA. And the chromosomes allow for compacting the DNA into the cell. And if you were to count up all the letters in one single cell of the body, you would find that there are 6 billion letters of information in one nucleus. If you were to look at how big is that? Well, one of the longest novels, not the longest, but one of the longest novels you may be familiar with is War and Peace, written by Leo Tolstoy. That only has about 3.4 million letters. And so the amount of information in a single cell is about 1,000 times larger than the book War and Peace. So as I mentioned, DNA is responsible for carrying that genetic information from parent to offspring. And as we saw in the picture, there really were equal contributions from both Reese and Ryan in the way that their daughter looked. And that's because exactly half of our DNA comes from our father through the sperm, and half of our DNA comes from our mother through the egg. So we are exactly 50% of each parent. Once those two come together, that sperm and that egg cell, we have all of the necessary information to create a human being. And so the DNA then tells the cell how to grow, how to multiply, how to form the eyes and the hands and the heart. And then after birth, those genes are also important in maintaining health. So exactly how many of these do we have? So when we talk about genes and chromosomes, the way to think about this, if we go back to our example of the book and say the book War and Peace represents all of the DNA in a single cell, then the chapters are represented by the chromosomes. And then the genes are represented by sentences within each chapter. So each sentence provides information, one bit of information, and that tells you how to make a particular part of the body. So there's a gene, for example, for eye color. There's a gene involved in height. There's a gene involved in hair color, so on and so forth. And so these genes are distributed across all of these chromosomes. And we know that in humans, there are 46 chromosomes. And because half came from mom and half came from dad, they actually come in pairs. And so we number the pairs from 1 through 22. And that's really in order of largest chromosome to smallest chromosome. And then the 23rd pair are known as the sex chromosomes. So in this case, we see an X and a Y chromosome. So we know this is a boy. If this were a girl, we would see 2 X chromosomes. Now, in the 90s, we wanted to figure out exactly how many genes were there in the human genome. And that spawned the Human Genome Project. And that took about 10 to 13 years to complete. And what we figured out is that there were about 21,000 genes in humans. So across all of these chromosomes. There are 21,000 genes. If you do the math, that's roughly about 500 to 1,000 genes on each one of these chromosomes. Obviously, if you're a larger chromosome, you're going to have more genes. If you're a smaller chromosome, you're going to have fewer genes. So how does that compare to other organisms? So just to give you an idea, the fruit fly, which is a familiar nuisance to all of us, has about 5,000 genes. So you might think this is a pretty simple organism. That's quite a few genes, but it's about 5,000. Corn, surprisingly, has about 30,000 genes, which is really shocking because it doesn't think, it doesn't move, it doesn't talk, and yet it has more genetic complexity than we do. And then the mouse, which is within our own family of mammals, has about 20,000 genes, very similar to humans, which is why mice are a really good way to study genetics and genetic disorders. So when we look at genetic testing, there's two sorts of things that we do in the clinic. The first is what's known as a chromosome analysis, and that's simply looking at the chromosomes, counting them, make sure they have no missing pieces or extra pieces, and confirming the correct number. The next sort of genetic testing that we can do is to actually read all of the letters that are in the DNA. So we might just look at one gene out of the 21,000 genes, depending on what we're concerned about, or we might do multiple genes. So an example of chromosome analysis, in this case, we see this happy, healthy little boy. And if we were to do his chromosomes, we would find that he has 46 chromosomes, so the correct number, and he has an X and a Y chromosome, as we would expect. Here we have this cute little girl in her adorable little outfit. But I think you might recognize features of Down syndrome in this kiddo. And if we were to analyze her chromosomes, what we find is instead of 47 chromosomes, instead of 46 chromosomes, she actually has 47 chromosomes. And if you look down here, you'll see that the extra chromosome she has is chromosome 21. And this extra chromosome 21 is actually what defines and causes Down syndrome. And you'll notice that 21 is actually a relatively small chromosome, which is why we do see Down syndrome as frequently as we do. Moving on to DNA analysis, in this case, what we do is we take our patient's DNA and we read through the letters, and then we compare what we see in the patient to what we call the reference genome. So when the Human Genome Project was completed, we said, okay, this is what looks to be the typical DNA sequence in the population. And so anytime we see a change from the reference genome, we call that a variant. And so this patient has a variant. The reference genome says that most people have a C at this particular position. In this individual, that is changed to a T. Now, it is very normal for people to have variants. That explains the great diversity and variation that we see in human beings. Most variants are completely harmless. They're just responsible for some of our physical differences. And if you were to look at one individual and compare all of their DNA to the reference genome, you would find about 4 to 5 million variants in one individual. And so it's those variants that make us unique. Now, the first thing that we often do in our genetics clinic is we go through a family history, because this might be surprising. With all of the advances that we've made in genetics, there is still nothing as helpful as a family history. There is so much information you can get from a family history. And we typically like to go through 3 generations. Sometimes we'll go further depending on what we're finding. But this is what we're doing. When we take a family history, we like to draw a pedigree. And a pedigree is basically a schematic used to identify relationships as well as individuals who are affected from one another. So in this particular pedigree, we typically use squares to represent males and circles to represent females. And a line across means that there was a union here. So this man and this woman had a union, and they produced a daughter. This daughter then had a union with this man, And between the two of them, they had 3 children. And I can tell that because of these vertical lines. They had a son, a daughter, and a son. This daughter had a union with an unrelated individual, and together they had 3 children. And then this son had a union and had 2 children through that union. And then what we do is we shade in individuals who have the condition that they are coming to clinic for. So in this case, we will use the example of a condition called polycystic kidney disease. This is a condition that's relatively common. It's a progressive condition where individuals usually begin presenting with cysts in their kidneys. Most of the time, they don't know about it, maybe in their late 20s or early 30s. But by the time they're 50 or 60, they begin to have declining renal function. So their kidneys begin having problems, and that's when the condition typically gets identified. So when we look at this particular family, what we notice is several things. So we're always looking for patterns. And what we see in this family is that the condition seems to be going across several generations. In this particular pedigree, 4 generations are affected with the condition. We notice that it doesn't seem to be skipping any generations because every generation has somebody who's affected. We also notice if we look at all the affected individuals, those are the shaded individuals, there's roughly an equal proportion of men and women who are affected. So there's 4 men and 3 women. And it also appears that the condition is being passed on at roughly 50% of the time. So for example, this woman who had 3 kids, 2 of her kids are affected, but 1 is not. This individual here had 2 kids, 1 is affected and 1 is not. So this is approximating 50% of the time it's being passed on. So when we see this kind of pattern, we know this is a dominant condition. And a dominant condition means that we have 2 copies of every gene. We get 1 from our mom and 1 from our dad. And apparently one of those copies has a change in it. And that change is dominant over the normal copy. And so just 1 copy being changed is enough to show the condition. So when we see a family history like this, we know that we have a high likelihood of finding a genetic change if we were to use genetic testing. And I just want to point out a couple of vocabulary words here. That was one of the things that came up in the questions. When we describe the condition, polycystic kidney disease, that is the phenotype. So the phenotype is what we can see in the clinic. The genetic change that we would identify, that's defined as the genotype. So genotype is what causes phenotype. Now, back in the old days, when we used to do genetic testing, it was very complicated. It took a lot of time. It was a very manual process. So in 1980, this is what DNA testing looked like. You might have seen this kind of a banding pattern before in some CSI episodes, for example. And using this technique, you could probably read a few hundred of letters of DNA sequence. By 1990, we had found a way to automate that process, and we were now able to get several thousands of letters being sequenced. And this is really the technology that we use for the Human Genome Project. It was a very long, laborious project. It took 10 years just to sequence 10 individuals. It cost $1 billion for the entire project. And so by the time we finished the project in 2001, it cost us about $100 million to sequence 1 person's genome. Now, as time goes on and new developments arise, the cost of sequencing a human genome steadily came down. And there is a law known as Moore's Law, which you may not have heard of, but you are familiar with. Moore's Law is a principle based in computing that says computing power will double every couple of years. And because of that change in computing power, the cost of producing things that depend on that come down at a predicted rate. And so Moore's Law would have predicted that the cost of doing a human genome should have dropped by this rate here. But you can clearly see that something happened right between 2006 and 2007 that dramatically changed how much it costs to do a human genome. And in fact, as of 2 years ago, we had broken the barrier of the $1,000 genome, and we are quickly advancing towards being able to do a human genome for $100, far different than the $100 million it took just 20 years ago. So what was it that happened in 2007? It was the introduction of a new technique known as next generation sequencing. I won't get into the details of it because it's quite complicated, but in comparison to what we were doing before, we essentially can analyze all of the letters in a human genome multiple times. And this is an example of what these next generation sequencers look like in our facility in New York. Now, it's very easy now to get the DNA sequence, but the challenge is how do you interpret what you find? So the first thing that's important is when we find a variant from the reference sequence, we have to classify this variant. And we've recently come up with five categories of how variants can be classified, but they really fall into 3 buckets. The first bucket is we found a change in a gene that we know quite a bit about, and we can predict that that change is going to cause a disease. So in that family with polycystic kidney disease, we likely would have found one of these kinds of variants. And we're able to use testing now to predict who might have the condition or be at risk for developing those cysts. The second bucket is what we call the benign bucket. This means we have seen this variant before. It's present in the healthy population. And so we think it's just it's just part of that normal human variation that we see. The third category, which is one that gives us the most heartburn, is something known as a variant of uncertain significance. And what this means is we found a change, but we've never seen it before in the healthy population, and we've never seen it before in patients who have a genetic condition. So we don't know how to classify this. And because DNA sequencing is relatively new, we are finding lots of variants of uncertain significance, which is why it's really important to keep in mind what is the reason behind why you're doing the test that you're doing. The other thing that's important is that our reference genome is changing as we are sequencing more and more individuals, and we're beginning to understand what's actually normal and what's not normal. But the issue is who is actually represented in those databases. And so if you were just to look at global diversity of DNA sequences that have been analyzed and that are in our databases, we will find a high representation from, say, European populations, but very low representation from Hispanic or African populations. And so a lot of work needs to be done for us to really understand the comprehensiveness of human genetic diversity. And I mention this because if you were to go out looking, can I get my genome sequenced? The answer is yes, anybody can get their genome sequenced. This is from a website of a company called Nebula Genomics. I have no affiliation or association with them, but I do happen to know that they are offering complete genomic sequencing to the general population. This is just a screenshot from their website. And they show you kind of the difference between what other companies like 23andMe and Ancestry DNA do, which is they look at just parts of the DNA. Nebula Genomics, on the other hand, actually does whole genome sequencing. But as I mentioned, we don't know a lot about what the DNA variants mean. And so if you were to get your genome sequenced, it may not be very useful at this point in time unless there's a particular question that you want to be, you're trying to ask. The second thing I would mention is that when I order a test in clinic, I have to order it through a lab that has gone through very strict certification. And that means that when they say there's a variant there, they are 100% certain that that variant is in fact present. Whereas direct consumer testing like 23andMe, they do not fall under those same strict guidelines. So sometimes they might make a call and they might say, oh, there's a variant at this position. But when I run that same sample in our clinical lab, we don't find the same change. So I would just caution you not to make any medical decisions based on direct-to-consumer testing. Make sure that anything you do find there gets confirmed in a clinical laboratory. So that's the kind of testing we use when we have a very clear evidence of a genetic condition running through a family. But what about if we were to see a family that looks like this, where there seems to be a lot of cancer in the family, but we can't quite see a pattern like we did in the other family? So for example, here we've got two brothers, one who had liver cancer and had a history of alcoholism, and then one who had prostate cancer, but he was relatively old, over 80. And then we have this person had a union with this woman who had breast cancer in her 70s, and she has a sister who had colon cancer in her 70s. And they have two children, one who had lung cancer but was a smoker. And now this individual, which is his son, is coming to the genetics clinic to find out, is he at risk for cancer? So this kind of pedigree is a little bit more challenging to analyze because it's not showing clear pattern. You know, it's lots of different cancers in different individuals. And what we know is when we look at overall cancer, about 70 to 80% of the time, cancers are sporadic, meaning they just arose by chance. They are not inherited. And then we see a small proportion of very clear inherited cancers, about 5 to 10%. These would include things like breast cancer due to the BRCA gene or the BRCA gene. There's also a colon cancer syndrome known as Lynch syndrome. So there's some very specific defined conditions that fall into this category. But then we have this sort of gray zone. These are what we call family clusters. And that's very much what this family is falling into. It seems like the cancer is clustering with this within this family, but it's not fitting any known pattern. So it may not be just one gene that's causing it. It might be multiple genes that are contributing. And we know about multiple genes for several other conditions. I'm going to talk about autism because I see a lot of that in my practice, and we've learned a lot about the genetics of autism. And what we believe is that autism is really a multifactorial condition with weak and strong genetic factors at work. But in addition to that, environmental factors play a role as well. And everybody has sort of this cup that they fill up with their genetic factors as well as their environmental factors. And once the cup gets full, then that's when we see autism presenting. So in this example, a couple of weak genetic factors, you can see those in green, and a few environmental factors, but those are not enough to reach the threshold to cause autism. Whereas in this individual here, we've got a strong genetic factor, 3 other weaker genetic factors, and 3 environmental factors that together all add up and cross that threshold, and this individual would present with autism. So here's another diagram just to kind of show this from a family standpoint, because we know these genetic factors can often be inherited. So here we have a father who does not have autism. He does have a weak genetic factor and some environmental factors, but it's low and it doesn't cross the threshold. Mom, on the other hand, has a pretty strong genetic factor, as well as 1 weak genetic factor, but very little environmental factors. So she also doesn't have autism. So the 2 of these individuals go on to have 4 kids, and 2 of them end up with a diagnosis of autism based on how things sort of fell out for them in terms of their genetics, but also what happened to them in terms of their environment. And environment is broad. Environment can mean exposures in the womb. It can mean events in life, whether they're traumatic events. It might be chemicals in the environment that we're not fully aware of. Those kinds of things all add up. So there's another condition that acts very similar to autism that has both genetic and environmental factors, but it has one additional component, and that component is time. And the condition I'm talking about is Alzheimer's disease. So Alzheimer's disease, just like autism, is due to a combination of both environmental and genetic factors, but those accumulate over a period of time. And it's this accumulation over time that is what ends up causing the condition. If you start off with a lot of genetic factors, then it's going to take less time for you to show symptoms of Alzheimer's disease. Whereas if you start off born with very few genetic factors, you may never show signs of Alzheimer's disease. So a few more details about Alzheimer's disease. When we look at the population, the vast majority of Alzheimer's, 75%, have no known cause. We think these are probably due to a complex interaction between genes and environment, but it's too complex for us to really understand it at this point. Then we have three other groups. We've got what are known as late onset familial. What that means is there are at least 3 or more individuals in the family with Alzheimer's disease, and their onset of Alzheimer's disease is sometime after the age of 60 to 65 years. And that represents about 15 to 25% of cases. The next group are what we call the early onset familial. Again, 3 or more individuals with Alzheimer's disease, but the onset is younger, less than the age of 60. This represents only 2% of all Alzheimer's cases, so it's quite rare. And then the last one is Down syndrome. We know that one of the genes that predisposes to Alzheimer's disease lives on chromosome 21, which is the extra chromosome that kids with Down syndrome have. And so if you have 3 copies of this gene instead of 2 copies of the gene, you might be able to, you're at more risk of developing Alzheimer's. And that represents just under 1% of cases. If we look at the late onset familial cases, the primary factor that we usually talk about is something known as ApoE4 allele. And you get one of these alleles from your mom and one of these from your dad. So if you happen to have both, if you got one from both parents, so you have E4 and E4, then there is a 30% risk of developing Alzheimer's disease by age 75. So that tells you that even if you have both copies of this particular allele, there's still a 70% chance that you're not going to develop Alzheimer's. So it's not a one and done. It's much more complicated. The genetics is only part of the picture. There have been several other genes, they're listed here, that have been associated with late onset familial Alzheimer's disease, but the risk that they provide is very low, maybe a 1 to 2% increased risk. So we don't routinely use these genes for testing because they're not very helpful. The early onset familial, however, is quite a bit different. These genes are have quite a strong effect. And the one that's the most common is this one known as PSEN1 or presenilin-1. It's found in somewhere between 20 to 70% of families who are affected with early onset Alzheimer's disease. The next gene that we see is the APP gene. This is the gene that lives on chromosome 21, which is why Down syndrome can have a risk for Alzheimer's. About 10 to 15% of families with the early onset form will have a change in this gene. And then the last gene is presenilin-2, accounting for about 5% of patients. So if you have an early onset family history of Alzheimer's disease, that's certainly an indication for possible genetic testing because there's a much greater likelihood that we would find something, and what we find might actually be informative. Now, doing genetic testing for someone who's asymptomatic but has a family history of early onset Alzheimer's disease is a challenging situation. It's not for everybody. Some people want that information, maybe to make preparations in their life. Other people might say, you know what, I don't want that information. It's not going to be helpful for me. It's just going to add to my anxiety. One of the other things, though, that people often ask is if I get this genetic test and I find out I'm at risk of Alzheimer's disease, could that impact me in some way? So it turns out that in 2007, we had the Genetic Information Non-Discrimination Act, also known as GINA. It was passed in the United States, and it provides protection for employment and health insurance. So basically, it says that employers and health insurance companies cannot use your genetic information to discriminate against you. So you have secured employment and you have secured health insurance. However, GINA does not protect against life insurance. It does not protect against disability insurance or long-term care insurance, which could clearly become important in patients who are concerned about something like Alzheimer's disease. It also does not apply to employers who have fewer than 15 employees. So if you're working for a small business, that protection does not extend to you. And it also does not protect US military service members. So those are some important things that we always talk about with families and patients when they're thinking about genetic testing, particularly if they're asymptomatic. So moving on, I've talked about some specific genetic disorders, ones that we know something about. But what about some of the really common diseases out there? Things like high blood pressure and diabetes and high cholesterol. Can genetics tell us something about those? So when genetics first came out, we really were focused on identifying single gene disorders, things like the presenilin gene for Alzheimer's disease, or the cystic fibrosis gene, or the sickle cell gene, or the hemophilia gene. All of them are caused by single gene changes. But things like high blood pressure and diabetes and high cholesterol, they're much more complex. They're caused by changes in multiple different genes. And so it's been really, we really needed the data science to advance so that we could really understand this better. One way of doing this is through something known as genome-wide association studies. I'll simplify it. Basically, what we do is we take a population, we find the individuals in that population. Here you can see they're colored and in blue and green. And these individuals, let's say, have high blood pressure. And then we take another population, those are the grayed out individuals, and let's say they don't have hypertension, high blood pressure, and we compare their entire genomes against each other. And then we pick out what are all the genes that are different between the two groups. We might not necessarily know the specific contribution of each one, but we know collectively all of these genes seem to be doing something in the group with high blood pressure that they're not doing in the groups without it. We can take this information and we can then say, let's take an individual from the population, look at their genome, and make a prediction about whether they might have a complex disease or not. And that process is known as a polygenic risk score. So say we're worried about coronary artery disease, we take an individual, these are their chromosomes, and we test them for variants in the genes that we identified from that previous research study that we did. And then we look at that pattern and we make a prediction. Where do they fall on this scale? Are they going to fall in a low risk category? Are they going to fall in a high risk category? Or are they going to fall somewhere in between? Polygenic risk scores are just now getting out there. They're not used widely for clinical purposes. There's a lot of research that's being done to sort of figure out how useful are they. If you were to do, say, a 23andMe, they use polygenic risk scores in some of the data they're providing, but there are a lot of problems with polygenic risk scores. For example, the accuracy of our risk prediction is highly dependent, again, on who have we analyzed before. And if we're analyzing a population that is not representative of the patient who we're testing in front of us, then those risk scores are not going to be valid. So this just brings me to this concept of diversity in genetics. It's really important because we can't really provide care for everyone across the spectrum if we don't have good information and good data. So I want to show something that kind of explains where our major problems are. So this is a photograph from the summit between the European Union and the African Union. And I'm going to just point out three individuals. This is Cyril Ramaphosa, the president of South Africa, Macky Sall, the president of Senegal, and Emmanuel Macron, the president of France. And there's something very interesting that happens if you look at the DNA of these individuals and you compare them to each other. It turns out that the DNA of Emmanuel Macron is much more similar to both President Ramaphosa and President Sall's DNA than if you were to compare the two of them to each other. And you might say, that doesn't make any sense. The two presidents are from the continent of Africa, and Emmanuel Macron is from the continent of Europe. Why does he have more in common with each of them than they do with each other? And that has to do with the evolution of human beings and the migration of human beings. So we know that modern man evolved roughly 300 to 400,000 years ago on the African continent. This is when we begin to see things like language, art, music, spirituality, all emerging within the human race. And then we advance over 300,000 years, we get lots of genetic diversity emerging on the African continent, lots of different groups. But about 65,000 years ago, a small group from the African continent migrated out of Africa into the Middle East and then up into Europe and into Asia. This group that left and populated the rest of the world was only a tiny sliver of all of the human genetic variation that existed at that time. And so what that means is that if you are trying to do DNA analysis and you are focused on one of these populations here, you're only going to get a small picture of what's true in our diversity. If we were to look how diverse is the population in our genetic databases, as of 2019, almost 80% were of European descent. Only 2% came from the African continent. So our picture of what is the reference genome and what is the normal genome is very, very skewed. We actually don't know all of the breadth of human variation. So this is something that we're really trying to change. What else can we learn besides getting a better reference genome by including diverse populations? I'm going to focus on just one of these bullets because there's lots of different reasons. It's this one right here, this second bullet here. It turned out that there was a genetic study being conducted in Africa on individuals who had low cholesterol. And when they studied them, they found that some of them had changes in a gene called PCSK9. And it turned out that changes in that gene caused your bad cholesterol to go down. When they learned about this, they realized that there might be a way to develop a medication that can use that information and be used to treat individuals with high cholesterol. And within 10 years of this discovery, we've had a whole new class of medications that are now used to treat high cholesterol. You might be familiar with something called Repatha. Repatha is a drug that is specifically used for this purpose. So there's lots of different benefits to having diverse populations being included in our genetic databases. I wanted to spend just a few more minutes talking about precision medicine, but also want to leave some time for questions. So just a couple things on precision medicine. One of the most advanced areas in precision medicine is something known as pharmacogenetics. This is basically how does your genetics impact how you respond to medications? We currently use a one-size-fits-all model. What that means is if all of these patients show up and have high blood pressure, we start them all off on the same medicine. And then if that doesn't work, we change the dose. If that doesn't work, we change the medicine. And we work our way until we find something. But you start off by giving the same medication. For some people, they respond beautifully and they don't have any side effects. For others, they end up either they don't respond or they have side effects, or they don't respond and they end up with side effects. And it turns out this is more common than you think. Roughly half of the time, a medication that's prescribed does not work for an individual. In the case of antidepressants, it's about 38%. In the case of Alzheimer's drugs, it's about 70%. Cancer drugs, it's about 75%. So can we not find a better way to do this? Well, it turns out that genetics actually impacts the way that you metabolize a drug pretty regularly. In fact, 97% of individuals, if they go through genetic testing of the genes that are important for drug metabolism, we can find a variation that impacts their particular medication choice. And we can group patients based on how quickly or how slowly they metabolize a drug. And so the hope is that instead of using this one-size-fits-all model, we would take people, test them first, and then pick the right drug and the right dose for them at that time. And then lastly, in precision medicine is cancer genetics. Cancer genetics has really seen a dramatic change in the last 20 years, mainly because we've started to understand the genetics of individual cancers. We used to classify cancers based on the organ from which they came. So if it's a cancer of the lung or a cancer of the pancreas or a cancer of the ovary, we would treat those based on the organ that they were coming from. But it turns out sometimes the genetic change in a cancer of the lung and a genetic change in the cancer of the stomach is the same genetic change. So you would actually use the same drug to treat both of those types of cancers, even if they're in two different organ systems. And so it's changed the way we think about cancer. We no longer think about it necessarily as it's a colon cancer, or it's a breast cancer, or it's a lung cancer. We're now starting to think of it as it's a this gene cancer, or it's a that gene cancer. And so this is just some of an example of some of the genes we're starting to associate with different types of cancers. So I think with that, I will go ahead and stop so we can have a few minutes to take some questions.
I am fascinated with the information. All I can think about is those 6 billion letters in DNA in one nucleus. And it reminds me of when you see a shooting star and it happened millions of years ago. It's just so vast that you can't even really comprehend that. I just applaud you for what you do. And just talking about how to do some type of testing for drug success. You know, there's so much that people go on, they try this, they try that, and the side effects are never good with pharmaceuticals and the interaction. So that just thrills me, actually. I have a question, the geno testing, and that scares me about the GINA stuff. I mean, I guess when you have life insurance, you have to go through the doctor and they have to know everything about you. So I kind of understand that. It still kind of scares me about Big Brother looking over. But with geno testing, how do you compare that to body scans? So some people I know have done these body scans that basically tell you what's going on completely in your body. Would you do that in addition to, or how do you compare those?
So in thinking about the differences, there's a little bit of overlap. So for example, when I talked about the polycystic kidney disease gene, we could test somebody for that in their 30s, but we could also just do a kidney ultrasound, because by the age of 30, most people, if they're going to have that condition, would show those cysts. So you could do either one. But what you can't do is you can't do an ultrasound, say, on a 12-year-old, because they won't have had cysts by 12. But the DNA change will already be there because it was there at conception. So we definitely are very careful when we use genetic testing, particularly in children, if it's not going to impact their health as kids, because we're going to take away their right to decide for themselves if they wanted to have that genetic information.
Okay, and a question just came in about Farr's disease, and I have one that was submitted at registration and one that's in the Q&A. How and where can I get tested? They actually said in the DC metro area. I don't know if you can answer to that. Strong family history and calcifications.
Yeah,
I don't
that's
know what Farr's disease is.
Yeah, so it's a condition where individuals develop calcifications within the brain. There is a genetic component to it. It's somewhat similar to Alzheimer's, where some patients will have a genetic form and some patients won't. But I think the proportion who have a genetic form is much higher than it is for Alzheimer's disease. So we do normally see patients for Farr's disease, and we offer testing to figure out, is it genetic or is it not genetic? In the DC area, the predominant group that there's several actually in the DC area. Most geneticists like me, even if they're part of a children's hospital, will see kids and adults. So Children's National Medical Center has a group of geneticists there, very large group actually. And then nearby is also the National Institutes of Health may have a program, and then Johns Hopkins, which is in Baltimore, also has a very robust genetics program. But if you're primarily in the DC area, I think I would reach out to Children's National. It might seem odd as an adult calling a children's hospital, but you'd be surprised. They do lifespan as well.
And someone just chimed in here. Yes, you will get the slides. Everyone will receive a copy of this webinar that I encourage to share with friends and family. But they also asked, do you know of labs that do this pharmacogenetic testing?
There are several labs out there that offer it. A couple of them are GeneSight and Genomind, just examples. The current way that pharmacogenetic testing gets done, if it gets done at all, is if you have an adverse reaction to a drug and your physician is struggling to try to figure out what to do next. Sometimes they'll either order it themselves or they'll refer to genetics and we'll order it in our clinic and then get the results back to them to help guide their next steps. The way that pharmacogenetics would work in optimally would be to do it before you is that we don't do really preventive genetic testing. It's only on the back end after you've had treatment failure that the insurance will then pay for it. So that's something that we have to find a way to change.
That's so unfortunate because we would end up saving so much time and money and health if we could do it on the front end and be proactive. And Mary just asked a question, is there much information about FTD?
Oh, that is a great question. So FTD is a frontotemporal dementia, and it's a condition that sort of has some overlap with Alzheimer's disease. There are more genes that have been now associated with FTD. So when we see patients for dementia, if it's depending on the information from the family history, from the neurologist who has been doing maybe the imaging studies, we decide on the best panel to try and capture all the potential genes so that we don't miss something genetic. Sometimes we pick panels that cover both Alzheimer's and FTD. Sometimes we just focus on FTD if the family history is more consistent with that. So yes.
So really the advice is to really look at the family tree a little bit deeper because she's concerned about her grandchildren, you know, that will be having children.
The most important questions that we often ask is not just who was affected, but when did their symptoms start and how quickly did it progress? Because those patterns tell us which genes are more likely.
And then I have a question. My father actually passed away from Alzheimer's, and I did not do the APO4 gene testing because I thought I can't do anything about it. Why would I want to know if I have a healthy lifestyle and I'm doing the things you're supposed to do, like eat well, exercise, sleep, meditate, you know, be social.
Yeah.
But now
you're controlling all those environmental factors that you have control over.
But I'm convinced that part of the contribution of my dad's Alzheimer's was he was a big weed guy and Roundup was like in his arsenal all the time. So I think it was definitely a lot of environmental. But now with these new drugs out, like lecanemab and Leqembi, do you change your thoughts on that?
Yeah, there are currently clinical trials underway to see if you find individuals who have ApoE4, and if you start them on some of those medications early, you might be able to delay onset. So we need a little bit more data to know if that's going to be the case. But if that does get proven to be the case, then I think it's another factor in the consideration to be tested. It should always be a patient's decision whether or not they get tested. Right. But they should be given all of the relevant information.
So the genetic testing that we were talking about, is that covered under insurance if it's prescribed or referred by a physician?
Yeah, these days it's pretty easy to get genetic testing covered.
Okay. Well, this has been amazing. I mean, I have so many more questions, but I love what you talked about, the unknown variant. that when people sign up to do these genetic testings that they can get through the mail, that really be aware that there's those unknowns. So you might be not getting as much information as you need, and it might be scarier than you need to know.
Absolutely.
I guess I'll end on that, but I cannot thank you enough. And remember, everyone will receive a copy of this. And that was the most informative. I mean, I loved when you were talking about the book chapter sentence, it just broke it down to make it so easy to understand. And of course, now I have 3 children. I'm going to be dissecting all of their, just like with Reese Witherspoon, dissecting all of their features and figure out who has more of what. But that was just a wonderful presentation. I can't thank you enough. And thank you so much to our audience for joining us today, and have a safe evening or afternoon, wherever you are. Thank you.
My pleasure. Thank you.